Hello, welcome to this new Journal of Physics video abstract. We are here at UPMC, Université Pierre et Marie Curie in Paris, and I'm talking to you from the Laboratoire Jean Perrin, where we're a group of physicists and physicochemists working on various biological systems. Through this short video, I would like to invite you to discover a paper about the impact of antibiotic treatment on the mechanical properties of the biofilms. We are talking here about biofilms formed by bacteria on surfaces. They are very heterogeneous material, as you can see on this three-dimensional reconstruction of a biofilm microvolume, and the big challenge here is going to achieve a relevant description of the mechanical properties of this heterogeneous material. To this purpose, we search for an experimental approach enabling to achieve local determination of the mechanical properties. Then we chose the route of the magnetic particles, motivated by the possibility to remotely actuate them, as you see here in glycerol. So here is an image of magnetic particles in red dispersed in a biofilm of GFP coli. Now, to really get local mechanical probes, we need to find a way to actuate them. To do this, Olivier Galy, here in the lab, has built these magnetic tweezers. They generate forces in the order of hundreds of piconewtons at the very heart of the biofilm, without altering its organization. Now, to measure the local mechanical properties, we are going to collect the stress-strain relation for each particle in the biofilm. During the magnetic actuation, we image the particles' trajectories in fluorescence at 30 Hz. You can see two different examples of this process on the slide. Then, from the particle displacements and the applied force, we derive the local viscoelastic parameters using the mechanical model of Berger. At the end, we get the map of the biofilm mechanical properties, as you can see on this representation, where the blue beads report the rigid environments and the red ones report the soft ones. Now we have a detailed reference mechanical profile and we can examine antibiotic effects. So we infuse the antibiotic in the nutrient flow at defined concentration for a given time lapse, and again, we collect the local creep curves using the same particles than for the reference biofilm. On this graph, you can see the relative differences of elastic compliance obtained for an ofloxacin concentration of 50 microgram per ml. In gray, the results obtained with the control treated the same way in the absence of antibiotic. Obviously, the 12 hours antibiotic infusion has induced very limited changes. Treated and control samples are very similar, and the small changes simply correspond to the slight evolution of the biofilm mechanics over 12 hours. At this point, it was important to know what had happened to the cells in this situation. Then, we tested cell viability using propidium iodide, a red cell death fluorescent marker. Clearly, the antibiotic-treated sample, which corresponds to the upper right image, exhibits a high percentage of dead cells, while only a low mortality is observed on the control. We have completed this investigation by testing another antibiotic which targets the cell membrane, while ofloxacin affects cytoplasmic enzyme function. And we have also examined a proteolytic enzyme that cleaves extracellular proteins into short peptides but does not affect cell viability. Please see the results in the paper. You will find there all the experiments supporting our main conclusions, which are that antibiotic treatments preserve cell shelter if not cells, and that targeting extracellular matrix might be more efficient than antibiotics to eradicate biofilms. Enjoy your reading. Thanks.